So my name is Alekhya Reddy, and my project kind of takes on a different format than the previous two, but I was looking into leveraging different cancer mechanisms for regenerative purposes. The first thing we're going to do is like a little personal introduction. Then I'm going to go over some of the cancerous mutations that can be used to increase rates of reproductions in cells. Then I'm going to go into how cancer has different metabolic pathways than normal cells and how mitochondrial function in normal cells can be increased. So my name is Alekhya Reddy. I'm currently a senior and I'm located in San Diego, California. And I was mentored by Divya B. And I chose to research this topic because in ninth grade, I was exposed to the history of HeLa cells. And I became really fascinated by cancer cells and then also just a personal background. So there is someone in my family who has kidney failure, and I just thought about the potential intersection between cancer cells, which are known for their incredible reproductive properties, and creating new organs or new tissues at a faster rate. So just for a quick overview, reproductive medicine has come far in the past couple of years, but the greatest limiting factor is the rate of cell growth, and it can take organs even the size of a fingernail months or years to grow. However, cancer cells are known for their incredibly fast rates of turnover, which occur due to mutations in specific genes and metabolic pathways in those cells, in addition to enhanced mitochondrial function. So implementing these mutations and enhancements in normal cells to be used for regenerative purposes could essentially change the game for regenerative medicine because we could have things be generated a lot faster. So the first thing I'm going to go over are the mutations, and mutations are abnormal changes in the DNA of a gene. So the first mutation is a pretty fairly well-known cancer mutation, and it's called IGF-1. And that is a gene that encodes a peptide growth hormone, and it's typically associated with an increased risk of breast cancer. And it is found in most stromal and epithelial cells, which usually line organs. And it tends to be a regulator of cell proliferation, which means cell reproduction, differentiation, which is when cells get their own unique jobs, apoptosis, which is cell death, and it's typically upregulated in cancer cells, which means you find more of it in cancer cells. The next mutation is acetylcholinesterase, and this is a signaling enzyme, and it's usually found in neuronal cells as part of a signaling pathway, the synaptic pathway, but in non-neuronal cells, it's involved in cellular growth, apoptosis, and drug resistance pathways. And so in cancer cells, you can find this pathway a lot of the times downregulated, which means you'll see it producing less acetylcholine. The next mutation that a lot of cancer cells tend to have is the MIR506 mutation. So MIRs are microRNAs, which are small non-coding RNAs, and they regulate gene expression. So this microRNA in particular regulates gene transcription, which is when the DNA of a cell is converted into an RNA sequence. And it's either an oncogene or a tumor suppressor, depending on which kind of tissue it's in, and it's found in all tissues in the body. So depending on whether it's an oncogene or a tumor suppressor, it can be upregulated or downregulated. And then the last mutation is TGF-beta, which is a cytokine, which is essentially like a protein that inhibits cell proliferation as a tumor suppressor in the gastrointestinal region. However, outside of the gastrointestinal region, this cytokine is actually found to be reversed, and it actually promotes growth by disrupting CDK inhibitors, which are essentially cell checkpoint inhibitors. And so for this reason, it's either downregulated or upregulated depending on which region of the body it's in. So these are just some diagrams showcasing how the first three mutations actually work in cells. So with insulin growth like factor 1, you can see how these insulin receptors lead to signaling pathways. And in combination with hormones, you get this TOR kinase signaling, which leads to cell proliferation. So increasing insulin like growth factor 1 essentially increases this whole pathway and causes more cell proliferation to occur. With the AKE pathway, which is typically downregulated, you'll see that it often leads to apoptosis or cell death. And you can see that it inhibits these three genes, which lead to cell growth. So downregulating this gene would mean less inhibition of these three genes, which would mean more cell growth, and it would create less of this gene, which would create less apoptosis. And then we can just see microRNA 506. So this is in the case where it's upregulated. It would increase cell growth by increasing the presence of these genes. It would inhibit apoptosis because it would decrease the presence of these genes. And there would be upregulated migration and invasion of cells and chemoresistance, which is just chemical resistance by increasing the presence of these couple of genes. So the next thing that is very different in cancer from normal cells are the metabolic pathways in cells. And metabolic pathways are essentially a linked series of chemical reactions that occur within the cell. So there are two main metabolic pathways that cancer reprograms. So the first is the glucose metabolic reprogramming pathway. So the PI3K AKT mTOR pathway essentially imports nutrients into the cell, and it's involved in the regulation of cancer cell growth, proliferation, and survival. And basically what it does is it allows for anabolic processing to occur even without growth factor stimulation, which essentially means that the glycolysis cycle keeps on going, which means that more energy is provided to the cell. And it's altered by certain components of the PI3K complex that increase energy production and anabolic processes. The second metabolic pathway that cancer upregulates is amino acid metabolism. So amino acid metabolism is closely connected with the glycolytic pathway, which is the conversion of sugar into energy, because it generates components of the cell cycle that are needed for these energetic purposes. So what cancer cells will do is they will use amino acid pools to fulfill the energy requirements for glycolysis instead of resorting to only using glucose. So that basically means there's an upregulation of glutamine transporters and production, and this essentially results in more glucose being converted into energy. So this is just a diagram of the glucose metabolic reprogramming. We can see that there are certain parts of the program that lead to cell cycle progression and cell proliferation, which essentially means that cells will differentiate and they'll reproduce more quickly. And we can see it also leads to increased mRNA translation, which means more RNA is converted into DNA for cells to do their job, increased lipid synthesis, and increased nucleotide synthesis, and a decrease in autophagy, which is essentially cell death. And this is just a diagram of what goes on in the amino acid pathway. So we can see that it's associated with one of the mutations I mentioned before, TGF-beta, which increases the presence of glutaminase, which increases the transition from glutamine to glutamate, which essentially provides the cell with the ability to undergo glycolysis and make energy from glucose. And the last thing that can be improved in normal cells is mitochondrial function. So the mitochondria is known as the powerhouse of the cell, and it's where cells generate their energy in the form of ATP. So increasing mitochondrial production increases ATP production, and ATP is what cells use as energy. So there's two key components of increasing mitochondrial function. The first would be increasing the amount of mitochondrial DNA, because mitochondrial DNA produces key catalytic subunits of the respiratory chain complexes that allow mitochondria to produce ATP. And modifying genes in the DNA can increase the amount of ATP production and decrease mitochondrial stress, which essentially leads to an overall increase in energy for the cell. There are also certain nutrients like vitamin E that help the mitochondria function, and increasing those nutrient availabilities will help increase mitochondrial metabolism by providing more fuel for the mitochondria and preventing oxidative stress, which means that ATP production will be increased as the cell is no longer under duress. So, just as a conclusion, the processes unique to cancer cells have immense potential to be used for regenerative medicine, and we can do that by emulating similar techniques in normal cells to increase reproduction rates of those cells. So, these implementations can be made possible by using gene editing technologies like CRISPR. Thank you. Are there any questions?
Thank you, Alekhya. Great job. Um, what are next steps for you? Are you looking to dive deeper into this topic?
Yeah, so the next steps would be actually like implementing some of these into human cells and seeing how they change the game for, increasing the production of tissues or building organs.
And are there any ethical concerns when talking about like implementing this in humans?
So, I mean, the most pressing ethical concern would be really knowing when to stop the genes that are increased or decreased so that the cell doesn't actually turn into a cancer cell. It just increases the rates of reproduction. So you would have to find that balance, testing just human cells before actually testing it on cells inside humans.